Hello and welcome. Today, we're looking at parasites in a new light. Often seen negatively, they are increasingly recognized as vital bioindicators, offering deep insights into the health and stress levels of our ecosystems. This field, known as environmental parasitology, studies how parasites and their host interactions reflect their surroundings. So, how do parasites act as sentinels of environmental change? The presence, absence, abundance, and diversity of parasite populations can signal various disturbances. Think pollution from heavy metals or pesticides, habitat alteration, climate change, or invasive species. Let's take pollution. Some parasites, like certain helminths, accumulate pollutants at far higher concentrations than their hosts or the environment, making them excellent accumulation indicators. Conversely, some pollutants can directly harm free-living parasite stages or ectoparasites, reducing their numbers. For instance, changes in water quality can make ectoparasites less common in polluted waters, while endoparasites might become more prevalent as host immune systems weaken. Acidification of water can reduce snail populations, which are intermediate hosts for certain trematode parasites, thus impacting their presence in fish. Environmental stressors, including pollutants and malnutrition, also weaken a host's immune system. This immunosuppression can make hosts more susceptible to parasitic infections, leading to higher parasite loads. So, an increase in certain parasite infections can indicate a host population under stress from environmental factors. Many parasites have complex life cycles involving multiple hosts. Their presence and abundance reflect the ecosystem's biodiversity and food web integrity. If an intermediate or definitive host population is affected by environmental changes, their associated parasites will also be impacted. A decline in a specific parasite might signal a decrease in one of its necessary hosts. Habitat alteration and climate change are major stressors too. Rising ocean temperatures and acidification, for example, influence parasite productivity and biodiversity. Temperature changes also alter the metabolic rates of both hosts and parasites, potentially increasing parasite intensity. Interpreting these signals isn't always simple. While pollution often means lower overall parasite species richness, some groups might thrive. Different parasite types respond differently. For example, parasites with a single host in their life cycle may adapt better to polluted habitats than those with multiple hosts, as the latter depend on a more complex chain of host availability. Environmental changes can lead to either an increase or decrease in parasitism. Contaminants might suppress host immune systems, leading to more infections. Or, they could be lethal to certain parasites or eliminate intermediate hosts, leading to fewer infections. Generally, ectoparasite infections might increase with pollution, while endoparasite infections might decrease. Parasite load, the number and virulence of parasites a host carries, is a key metric. Stressed individuals often show higher loads, though if conditions become too severe and kill hosts, this could also limit parasite propagation. This means using parasites as bioindicators requires a holistic approach. We need a comprehensive understanding of their biology, life cycles, and interactions with hosts and the environment. Combining parasitological data with other environmental quality assessments improves accuracy. In essence, parasites are integral to ecosystems. A healthy ecosystem often supports a diverse and stable parasite fauna. Deviations from these natural levels, increases or decreases, can be valuable early warnings of environmental degradation. This makes parasite ecology an indispensable tool in environmental monitoring. The ongoing challenge is to refine our understanding of these complex interactions, allowing us to effectively use parasites as reliable sentinels of ecosystem health. Thank you for listening.